This is lecture 6 from chapter 15, and this is viruses, the last of the microbes. Very big topic today, obviously, with COVID-19. And so we're going to be looking at what a virus is, how it infects, and what kinds of things we can do to protect ourselves. So the first thing to look at is why viruses aren't actually a living organism and how they are structured. So first, let's just take a look at the basic structure of a virus. So all viruses have two things. They all have genetic material. Now, the difference here is when we looked at eukaryotes and prokaryotes, the genetic material was DNA. But as you can see here, the genetic material is DNA or RNA. So you can have a DNA virus, you can have an RNA virus. So we have those two different groups. So it doesn't have to have DNA as its genetic information. The other thing that all of them have is this capsid. The capsid is basically the container or the wrapper for the genetic information. Those two items, these two right here, are the absolute minimum you need to make a virus. That's it. Think about it. When we talked about a prokaryote, you had to have a cell membrane, the wrapper, and you had to have genetic information, but you also needed to have things like a ribosome. You had to have ribosomes to make proteins. You needed to have cytoplasm. You needed to have those other materials in order to make a bacteria. These are the only things you need to make a virus. Now, sometimes those viruses are going to have an envelope or a plasma membrane outside of the capsid, and that's what you see here. That's just an option. And those envelopes will have glycoproteins. We'll talk about and see these later on and why they're important. So you can have two different groups. You can have the envelope viruses. These are the ones that have the plasma membranes on the outside, so they have those wrappers, or the non-enveloped viruses. So these are the ones that just have, you can see that capsid. They have those shapes. So those are the basics of what a virus is. So this is, like I said, a big reason why these aren't considered alive, because they don't have the machinery to make proteins. They don't have the machinery to take food like sugars and break it into ATP and energy. They're basically just genetic material containers. The other thing is, if you think about something that's alive, is that it has the ability to reproduce. And viruses cannot do this. They actually have to have a host in order to have reproduction happen, and they can't do it on their own. So let's take a look at how viruses reproduce. So here is how you get a new virus. So the first thing that happens is you get a virus that binds to the cell membrane for a host, and then the viral DNA gets pulled into the host cell. Sometimes the whole virus might get pulled in, but this is the important part right here, getting the viral DNA inside. Now you can tell that this is a eukaryote because we've got the nucleus, but this can happen to prokaryotes too. With the prokaryote, the DNA would just go straight to the chromosome. Here, the DNA is going to be taken into the nucleus, and they don't show it here, but the DNA is going to actually be put into the chromosomes of the host organism. Then we're going to have the cell itself take that viral DNA, and it's going to make copies of it. So it's going to use the cell's own energy, own materials, and it's going to have it make copies of its DNA. That DNA is also going to contain information on how to make mRNA. And so it's going to transcribe mRNA. And if you remember, we're going to use mRNA to make proteins. And so that mRNA is going to get shipped out to the cell, to a ribosome, and that mRNA is going to be read to make the proteins for the capsid. So now we've got the virus has gotten its DNA transported in. The DNA has been copied. So we've got new copies of the genetic material that will get sent out into the cell. We've gotten mRNA made. The mRNA is read and we get viral proteins made for the capsid. And then these guys self-assemble. So the capsid will snap around individual DNA strands, as you can see here. And then most of the viruses will actually just migrate out of the cell. Some viruses actually will not be able to migrate out like this, and what they'll do is they'll just fill up the cell until the cell explodes. But the big thing here is that they're dependent on the hosts for their machinery and for the energy to make new viruses. And so this is the other big reason why we don't consider these guys to be alive, because they have to hijack a cell in order to reproduce. And they don't even do the reproducing. They get the cell to do it for them, which in a way, if you think about it, is kind of cool. All right. So how can we protect ourselves then? Because remember, bacteria, we can use antibiotics, but antibiotics do not work on viruses. So the big way we can protect ourselves is for a vaccine. So how are vaccines made? Well, I'm going to look at flu vaccines. So flu vaccines, these are actually how the old flu vaccines were made. Because most of them were egg-based. The newer ones are actually cell-based. It actually works a very similar way. So I'm just going to walk you through. The first thing that has to happen is that the CDC has to decide which strains are most likely to infect us in the next year. So they're going to pick 4 strains that they think is most likely going to make us sick next year. Then what they're going to do, if it's an egg-based vaccine, they're going to inject fertilized eggs with inactivated or weakened live strains of that virus. Because what they want is they want these to actually infect the eggs so that the egg cells will start manufacturing copies of the virus. Now, I mentioned that not all of these are egg-based now. They're cell-based, and they do the same thing. They're going to infect cells that they have in cultures to do the same thing. So then the cells are going to reproduce the virus, which is what we want. We want lots of copies of the virus. And then we're going to take the fluid that contains the virus, collect it, purify it so that we can get, as just the virus, and then we quote kill it. Now, we just said that they're not alive, so we're going to kill it. Basically, what we mean is we're going to inactivate it. So there are different ways to do this, using things like radiation, heat, and so forth. What we usually want, and this is what we want with the flu virus, we want the capsid to stay intact, but we want the DNA to be scrambled. We don't want the DNA to no longer be functional to cause problems and be copied. But we want the outside to be there so that our immune system has something to react to. So why vaccines work, I'm going to post this video. Please watch this video because I will be asking you questions about it. I will talk about it. But this explains how the vaccine actually works once it gets into your body. So you can see how it works and how effective they have been. So once you get done watching that video, come back, watch, and I'll pick up with why can't everyone get vaccinated in our next lecture.